well as from euplotis adiculatus. And the quality of the RNA was checked by running it on a denaturing gel electrophoresis, and 2 bands were observed, as you are seeing. And from this RNA, we have prepared the cDNA, and we have checked this by performing the PCR, and we did the qPCR to do the quantitative assay, that is whether there is increased in the amplification is there, and we did the real-time PCR to know about the expression of these genes, that is expression of again MNSOD gene, catalase gene, and HSP70 gene. Hope I am clear. Everything is clear so far?
Yes, ma'am. Yes, ma'am.
So ciliates were exposed to LC50. The dose which we selected for the expression of stress responsive gene was that LC50 dose for 24 and 48 hours were selected to evaluate the expression of stress responsive genes, that is MNSOD, catalase, as well as cytosolic HSP gene. So experiments were performed in triplicates, and there was a significant increase in the expression of stress responsive gene in both Tetmemna sapri as well as in euplotis adiculatus. So first gene that we have chosen is MNSOD gene. It's an MNSOD gene, and it was amplified using the custom synthesized primers and using the customized synthesized primer, and maximum expression was observed after 48 hours of exposure of metal, cadmium and copper, that is in both the ciliates. Thus, this could be because MNSOD is expressed after copper zinc SOD. Actually, there are 3 types of SOD present in the cell, and MNSOD is one type of SOD present in the cell, and it gets activated after copper zinc has activated, and this MNSOD is reported to last longer duration because it has a tetrameric structure, and it has been found to have more number of metal responsive elements or antioxidant responsive elements on the promoter region as compared to copper zinc SOD. And we also studied the activity of expression of catalase gene, and as you can see from the slide that the expression of catalase gene has also increased. We studied the activity of expression of HSP70 gene. Even the HSP70 gene, you can see that there is increased in the expression of HSP70 gene after we expose them to LC50 dose 24 and 48 hours. Okay, so now our next thing was to characterize these genes. So far, no one has characterized these genes, though their expression was going up, we could see that, but we had to characterize these genes. So MNSOD was amplified using custom synthesized primer, and the amplicon size of MNSOD gene was 585 base pair. Okay, and it was submitted again to the gene bank with the following accession number, and blast result of protein sequences showed that it is 86.39 similar with the Stylonychia lemnae and 85% similarity with Oxytricha triflex. The protein sequence of MNSOD contained high content of leucine residue and low content of methionine and absence of cysteine residue. If we look at this slide, you will find that there is a signature sequence which was identified at a position between 163 to 170, and the predicted 3D structure of an MNSOD in the case of Tetmemna sapri that it is a homotetramer in which each monomer contains one active site. An active and catalytic site was further analyzed by using the Swiss model and iTesser online server, which contained Mn2 plus ion at the center, surrounded by 4 important amino acids like histidine, that is histidine and aspartic acid, that is 3 residues of histidine and 1 residue of aspartic acid. And in this study, for the first time, MNSOD was sequenced and characterized in the freshwater ciliate. Next gene that we characterize is catalase gene, and the gene was again amplified using the custom synthesized primer, and the amplicon size was 420 base pair coding for 140 amino acid. The partial sequence of CAT gene of T supply was again submitted in the gene bank with the following accession number as given here. The blast result showed its similarity of 77.78 similar with the cat gene of Tetrahymena thermophila, and again the protein content has high content of alanine and lysine and low content of arginine, and there was absence of cysteine residues. Again we did the protein sequence characterization, and it shows more similarity with the monofunctional catalases belonging to the clade 3. Actually catalases belong to 3 clades, clade 1, clade 2 and clade 3. The one which we have purified and characterized belongs to the clade 3, and the predicted 3D structure of catalase protein in both the ciliates were similar to the other reported CAD protein. The predicted 3D structure, as you can see from the slide, again it is a homotetramer. The catalytic site was analyzed again using Swiss model and iTesser as well as Autodoc software. The catalytic active site had heme group, which is known to bind to hydrogen peroxide or other xenobiotic compounds and facilitates catalytic reaction by breaking down H2O2 into water and oxygen. Okay, so the structure is homotetrameric with 4 identical subunits arranged tetrahedrally, each containing heme group and an NADPH at the active site. Iron ion in the heme group is known to be involved in catalyzing hydrogen peroxide into water and oxygen. Now this is the characterization of catalase gene from Euplotis adiculatus. Again it was the amplicon size was 858 base pair, and it was again submitted in the gene bank, and it showed similarity with Euplotis vanus. And the predicted 3D structure of catalase protein shows again that it is a homotetramer with heme present as prosthetic group at all the 4 sites. Now what we did was we wanted to know, can we compare the catalase gene of the 2 species which we have with the known group which is more closest to this, and the most closest group which we found was Tetmemna species SEG215, whose genome was already submitted to the gene bank, and we compared using the bioedit software version, and we found around 45 amino acid variation was observed in Tetmemna SP and Tetmemna SCG, and out of this, so it shows there is 9.26% of this similarity between them, whereas 79 amino acid variation was observed between Tetmemna SP and Euplotis adiculatus, which shows that there is 16.26% dissimilarity between the sequences, and active sites were analyzed in all the 3 species. That is 2 of ours and the one which was most similar to our strains. We found that the active site was analyzed, and it was observed that there are 3 main conserved catalytic amino acid residues, that is histidine, asparagine and tyrosine. Similarly, in Tetmemna sapri, the active site contained histidine and asparagine residues, showing that the active site are conserved in both these Tetmemna species, whereas in the active site of Euplotis adiculatus, we found, in addition to histidine and asparagine, in lieu of asparagine, we found lysine. So there are variation if you have a level which we could detect by making such a kind of study. So this is characterization of the catalase gene in all the 3 species. Now last thing that we did was, sorry, I will finish within 2-3 minutes. The characterization of HSP70 gene. The partially sequenced cytosolic HSP70 was submitted again to the gene bank, and it was found to have similarity with the cytosolic HSP70 gene of Sterkiela, that is Oxytricha nova, and to Tetmemna. The sequence was that it was around the length of the sequence that we could sequence was 1758 base pairs, which was encoding a polypeptide of about 586 amino acids, and it was found that it has this protein has got lysine was present in high content and tryptophan content was low. And protein sequence, as we could see from the next slide, that if we characterize the gene, there are 2 basic regions. On the N-terminal domain we have ATP binding domain, and on the C-terminal side we have substrate binding domain, and this ATP binding domain is conserved across. And in fact this work is also published in 2020 and 2022, both from Tetmemna sapri as well as from Euplotis adiculatus we have published this work. And now to conclude the whole thing, these ciliates can be used in ecotoxicological studies. Why? Because they are economical. They are very simple to culture in the lab, and they provide a benchmark to monitor the extent of ecological damage induced by anthropogenic activities. Since I have already told you that Yamuna river is contaminated, we have collected the samples from Yamuna only, and we could detect these ciliates. It means they are surviving in those harsh conditions, so they must have some cellular molecular tool which is helping them to survive. So in both sapri, Tetnamna sapri and Euplotus ediculatus, we can use them as a model system for assessing heavy metal toxicity. Why? Because you can use the following endpoints as to use them in the study, that is effect on the morphology, then effect on the antioxidant enzymes, and their effect on the stress responsive genes. So this study affirms that the above mentioned endpoints are suitable biomarkers for monitoring the heavy metal toxicity. That is, you can use antioxidant enzymes or you can use HSP70. They will serve as a good biomarkers to mediate to study the cell mediated stress response in these ciliates, and these can serve as a model system for ecotoxicological studies. So before I conclude, I must acknowledge our principal, Acharya Narendra Dev College, Professor Ravi Tuteja, who is my co-presenter also for the present study. And this work was supported by a project from UGC and a project from DST. And presently the lab is running a barcoding project sponsored by DST sir. And these are our contributors. This work was mostly done by Dr. S. Shripurna, who is presently working in OYO University. Dr. Jiva was there. Swati, Sandeep, and Jyoti are presently working with me as the PhD students. I must thank all of you for helping me in making this presentation. I must thank the organizers also for giving me this opportunity to deliver the talk at this international platform. Thank you so much.
Thank you so much, ma'am, for your inciting full lecture. Your presentation was highly informative and covered valuable content.
Thank
Despite your busy schedule, you graciously took the time to share your expertise on very short notice, showcasing your dedication to research. Thank you so much, ma'am. Thank you so much.
Thank you, thank you, thank you.
Our next international guest speaker, Dr. Giridhar Babu Anam, is an assistant professor in the Department of Civil Engineering at Yangam University, South Korea. Uh, He received his PhD degree in 2011 from the Department of Biotechnology and Environment Science, Thapar University, India. He has been worked in various National University Institute, Combac National University, Kongong National University, Postec, and has experience of about 11 years in teaching and research in the area of environmental microbiology and biotechnology in Korea. His research scope encompasses the application of biochemical agents for the phytoremediation of soil stained with inorganic pollutant as well as bioremediation of hazardous substance releases into aquatic environments and microbial metagenomics. Currently, his research focus lie in evaluating the hazards of cyanobacterial blooms, exploring chemical and cyanophage facilitated eliminated methods. He is an author and co-author of several international peer-reviewed journal, book chapter, and referred conference paper. He has participated in several national and international research and monitoring innovative and served as a reviewer for several international journals book. Now I would warmly welcome to Dr. Giridhar Babu Anam sir. Please sir.
Am I visible?
Yes sir, very much visible.
What about my slides? Is are visible
Yes,
or
sir. Yes, sir.
okay?
Visible, visible.
Okay, thank you very much. And first of all, I would like to thankful to conference committee for inviting me to deliver my talk on fast therapy for sustainable management of cyanobacterial harmful algal blooms. And these are the contents to be delivered in this presentation. And freshwater ecosystems under seas and cyanobacteria and harmful blooms, cyanotoxins and health hazards, risk assessment of cyanoblooms and cyanotoxins, and how to combat these cyanobacterial blooms from the aquatic ecosystems and as well as wastewater treatment plants. And the last one is the main topic is it covers the cyanophos technology, advantages and challenges. Sir, my slides are moving?
No, sir. No, not moving.
Not moving. Okay. Now?
No,
Now,
this is first slide only.
first slide.
Yes, sir. Now moving.
Now moving, right? Okay.
Yes, sir.
Okay. So, first slide. Okay. Freshwater ecosystems. Freshwater ecosystem is nothing but any water columns. It should be like a low salt content. It making suitable for the any plant growth and animal growth. We can simply call it as a freshwater ecosystems. But thing is that nowadays freshwater ecosystems are indeed facing various threats and challenges. Among various threats and challenges, few are I'm going to consider to explain here. One is over extraction of water and dumping of waste into the aquatic systems. And another one is discharge of wastewater. And fourth one is climatic change. Among these threats, two are main factors involving in the freshwater ecosystem under siege. Those are discharge of wastewater and climatic change. So what is discharge of wastewater? As we know nowadays, there is a rapid increase in the population and population activities which results in establishment of huge industrialization. Due to this huge industrialization, there is a byproducts which are ultimately releasing into the releasing into the aquatic systems. And mainly the byproducts containing the nutrients, those are the very effective and they are negatively affecting the wastewater ecosystems. And the second one is climatic change. Climatic change due to global warming, which causes sudden changes in the temperature and the light, which are effective in the way of releasing the nutrients. Releasing the nutrients means nutrients are already existing in the water. These factors directly involving the availability of the nutrients for the microorganisms. Altogether, which causes the cyanobacterial species dispersal between the aquatic ecosystems. So, what is the cyanobacteria? Until now, we discussed the cause of the cyanobacterial bloom. So first of all, going to the blooms, what are the blooms and what are the toxic effects of the blooms? We should know about the cyanobacteria. Cyanobacteria is commonly known as autotrophic blue-green algae. It contains as usual like plants, chlorophyll, phycobiloproteins, phycocyanins, and phycoerythrins. These are the pigments which are useful for the photosynthesis and like other systems like protecting the photosynthesis like that. And another one is they have a wide range of cellular strategies and physiological adaptations. They play an important role in the ecosystem and food web maintenance. So generally cyanobacteria in the ecosystems exist in the two ways, two large ecological groups. They can exist in the two ways. First one is planktonic cyanobacteria. This type of cyanobacteria float freely in the water columns. And another one is benthic cyanobacteria. This benthic cyanobacteria generally stay in the submerged solid surfaces like sediments, rocks, stones, and aquatic plants. Currently, there are 5,323 species, 425 genera, 65 families, 13 orders. They are classified like a big cyanobacterial group in the water systems. Significance of the cyanobacteria. Generally, there are two types of significances. One is ecological and second one is economical. In case of ecological significance of the cyanobacteria directly involved in the carbon fixation, carbon cycle, and few species of cyanobacteria involved in the nitrogen fixation. In case of ecological importance of the cyanobacteria, they are very useful for the production of biofuels, cosmetic compounds, nanomaterials, recombinant proteins, and so on so. Apart from the ecological benefits, there are some cyanobacteria, they form blooms. What is bloom? A bloom is nothing but a cyanobacterial bloom is an overgrowth of cyanobacteria. Is nothing but overgrowth of the cyanobacteria. We can see in the any aquatic system, we can call it as a blooms. Under favorable conditions, they dominate the phytoplankton communities and form a dense layer, mats, scums, and blooms. In the recent decades, the occurrence of cyanobacterial blooms has increased temporally and spatially due to the anthropogenic nutrient over-enrichment and climatic changes. These are the few cyanobacterial blooms that can also produce the toxins in the water bodies. These are the few examples you can see in the slides. And not only producing the cyanobacterial blooms in the water system, they can also synthesize, they can also produce the cytotoxins in the body. After synthesizing in the body, ultimately that cytotoxins could be released into the water bodies that ultimately causes the illness of the human bodies, not only human bodies, any aquatic organisms like plants and animals, not only inside of the aquatic system, surrounding who are living, they are also getting infected with this type of these cytotoxins. See, generally, exposure sources of these cytotoxins are direct contact with the water. And not only direct contact with the water, who are residing surroundings of the ecosystems, they are also affecting with this type of the cytotoxins due to the airborne and aerosol capacity of these compounds. It has reported that 174 animal deaths and 458 human illness in the USA because of harmful algal blooms during the 2007 and 2011. Generally, cytotoxins are classified into three main phases. One is hepatotoxins, neurotoxins, and dermatotoxins. The name itself you can see here, hepatotoxins are liver damaging toxins, and the neurotoxins are they can work on the neuromuscular system, and the dermatotoxins are they affect on the dermatitis, I mean skin related problem. They cause skin related problems. If you can see, there are another type of the cytotoxins here. They can also causes the taste and odors in the water suppliers. For example, jasmine and 2-methyl isobromanol, 2-ionone, and beta cyclosystol. These are the compounds which cause taste and odors in the water supplies. If the compounds are present in the any water, you can feel this type of tasteless and odors in the water. The aggregated exposure pathways of the cytotoxins. If you can see in the slide, there are the limitation guideline value for these toxins present in the water systems. If you can find these levels of the cytotoxins in the waters, we cannot allow for the drinking purpose. For each compound of cytotoxin, there is a limitation we should follow while we are utilizing the any ecosystem water for the drinking purpose. Not only this one, you can also find the effective illness of these cytotoxins in the human bodies. You can see here few of the cytotoxins like microcystin LR, they're directly affecting on the liver. Another anatoxins, they are affecting on the brain. Like that, few of the cytotoxins, they are directly affecting on the human organs. So what we have to prefer here, we have to critically analyze the water before we are consuming. So what are the factors which are influencing the cyanobacterial bloom formations? There are presently two identified factors directly affecting on the cyanobacterial bloom formation as well as the cyanotoxins production in the water bodies. The two factors are biotic factors and abiotic factors. Biotic factors involving the organisms residing with the cyanobacteria. If you can see in the any aquatic system, cyanobacteria alone cannot survive here. They are associated with the number of other organisms. They can also promote the cyanobacterial blooms. They can change interlinking. They have the interlinking between them among them. They can change their food stuff each together. They can promote the each other also. So these are the abiotic factors which involve in the cyanobacterial biobloom formation. Another factor is abiotic factor. Another abiotic factor, there are a lot of things. They are also supporting the bloom formation. First one is anthropogenic eutrophication, and second one is urbanization, and third one is agricultural practice. And as usual, global climate change. Among them, we can mainly focus on the anthropogenic eutrophication. Because of this anthropogenic eutrophication, we can find the lot of water disturbance because of main reason is eutrophication. As we know, the eutrophication is a process in which water body becomes overly enriched with nutrients because overflow of the nutrients from the industries, various industries, without treatment, they're directly pumping into these waters into the aquatic system. Due to this problem, they're growing very rapidly in the water bodies. They're preventing the growth of the other organisms. Preventing the growth of the other organism means nothing but the deficiency of the oxygen for the other microorganisms, because cyanobacterial bloom forms in the surface of the water. Due to the forming of the surface of the water, light penetration will be decreased. The overutilization of the oxygen in the water, they cannot available for the other organisms. Only cyanobacterial blooms will be dominated on the surface of the water. So main biological traits of the bloom forming cyanobacterial cell. If you can see any bloom forming cyanobacterial cell, you just go and pick any bloom forming cyanobacteria, you can find the four things. You can easily observe the four observations there. They are actively participating in the cell metabolism. First one is gas vesicles, and second one is carbon utility and phosphorus acclimatization, secondary metabolites. Gas vesicles are generally it forms to beyond the cell blooms on the surface of the water. Carbon utility. During the bloom formation, the metabolic activity, I mean carbon fixation is very high. And third one is phosphorus acclimatization. If you can see the any cell under the microscope, you can find the phosphorus acclimatization. You can find the polyphosphate granules in the cyanobacterial cells. And fourth important one is secondary metabolites. You can see the lot of secondary metabolite accumulation. Secondary metabolites means there are many other secondary metabolites which are useful. But apart from that secondary metabolites accumulation, apart from that useful stuff, you can also the accumulation, high amount of accumulation of cytotoxins, which are very negative impact, which causes very negative impact to the aquatic ecosystem. So risk assessment of the cyano blooms and cyanotoxins, because we have to monitor every time, because we have to focus on the aquatic ecosystems, because many countries the water source is coming from the aquatic systems. So we have to always continuously monitor by using a different kinds of the techniques. Under this techniques, if you can see, first one is monitoring and modeling. Here we have to find the water samples, collecting the water samples and see the chlorophyll quantity, and by using different techniques like microscope, by using fluorescence techniques, we can go for the fluorescence analysis of the chlorophyll like that. And one more other technique is satellite remote sensing. We can also use the satellite systems to monitor the cyanobacterial bloom formation in the various aquatic systems. And one more one is the deep neural networks. There are a lot of modeling techniques nowadays available. We can go for the risk assessment of the cyanobacterial blooms and cyanotoxins. But here we can find by using these techniques only the cyanobacterial bloom formation, but we cannot go for the estimation of the cyanotoxins. For the estimation of the cyanotoxins and the identification, because about these techniques only you can see with your naked eyes, oh yes, there is some bloom formation, there is some grainer vision on the water bodies. These things only you can predict by using these technologies. In case of if you want to go for the depth knowledge, you have to use the other technologies, molecular technologies, to find the which cyanobacteria, which type of species are dominant there. For that purpose, we have to go for the many molecular techniques like PCR, RT-PCR, and qPCR, and many things. In case of the cyanotoxin monitoring, we have to go for the instrument analysis. Like in this instrument analysis, there are also some available kits like ELISA kits. You can directly pick the sample and put these reagents. You can find the cyanotoxins presence or not. Another techniques are there. You can directly quantify the amount of the cyanotoxins presence in the water bodies. Those are LCMS and some other techniques like gene confirmation techniques by using qPCR. Here you cannot predict the cyanotoxin toxicity quantity, but you can predict, you can analyze. Yes, there is some chances of the production of the cyanotoxins in the water body based on the presence of the genes. But here you cannot say that if the presence of the gene is there means it is directly proportional to the quantification, identifying and quantifying the cyanotoxins, because you cannot predict here. Just you can find, yes, there is a chance. Yes, there is a presence of a gene in the any organisms like cyanobacterial blooms, but you cannot predict that that gene can express and that gene can produce the like cyanotoxins like that. These are the current techniques. So this is the main thing here, main important slide. It can show something like how to combat the cyanobacterial blooms. There are currently two observations. First one is prevention method and second one is mitigation method. Prevention method, we know prevention is better than like you can say, without going for the mitigation method, you can go for the prevention method. Prevention method generally includes nutrient input reductions and early warming. Warning means time to time continuously monitor whether there is a cyanobacterial blooms are forming or not. Prediction modeling, we can find, oh, there is a chance of producing the cyanobacterial blooms now. So what are the solutions to prevent them? Those are the prevention methods are there. If you can go for that one, you can reduce the bloom formation at this different seasonal conditions. Another one is mitigation methods. In the mitigation methods, you have to follow the some treatment methods to remove the cyanobacterial bloom from the water instead of preventing the formation of the blooms. So there are a lot of techniques currently available to mitigate the cyanobacterial blooms from the water bodies. Now, I am going for the bloom prevention methods. First one is coagulation and flocculants. And the coagulation and flocculants is nothing but using some materials to precipitate or bind the available nutrients in the water bodies. Generally, we are using aluminum related coagulants which are aluminum sulfate, aluminum chloride, some of the polymers such as polyaluminum chlorides. We are using this type of coagulants to precipitate the available nutrients in the water bodies. This is the one type of prevention method. And second one is bloom control and mitigation method. Generally, we are following two types of method here. First one is physical or mechanical control, and second one is chemical control. In the physical control, we are using filters, pumps, some barriers. In case of the chemical control method, we are using some chemicals like magnesium hydroxide, some hydrogen peroxide, some copper related materials to kill the cyanobacterial blooms here. Anyhow, these are the good explanations. I don't want to explain here more, and I am directly going for the what are the advantage and disadvantages of above mentioned techniques like physical or chemical techniques to remove the cyanobacteria from the wastewater bodies. Despite the effects in specific conditions, yes, we have to agree that above mentioned techniques are somewhat is good, but they are time consuming, short lived, and cost also. And moreover, those type of techniques causing secondary pollution sometimes. And another one is negative impacts on non-target species in the water bodies. It is very important. We have to maintain the ecosystem. We are focusing on the cyanobacterial bloom to remove them from the water bodies. But instead of that, if we are using some of the toxic chemicals, they are also affecting on the non-target species. So alternatively, biological algal removal agents are gradually recognized due to their eco-friendly roles. However, some biological agents and their bioactive materials remain further tested and optimized. Altogether, we can say biological methods are very good, but still we have to test and optimize it. Among them, the more targeted and specific approaches should be investigated with fewer negative impacts on the ecosystem service. We have to see. We cannot find, 100% results, but we have to see whether they are reducing the negative impacts compared to the other treatments. We have to go like that way. In that way, we can go for the cyanophage technology nowadays. If you can see the cyanophage technology or therapy, before we discuss about the cyanophage technology, we have to know about the cyanophages. What are the cyanophages? Cyanophages are a class of viruses that specifically infect and replicate the cyanobacteria. What is cyanophage therapy then? Okay, we are saying cyanophages are a class of viruses that specifically infect and replicate the cyanophyte. What about the cyanophage therapy? Here, nothing here. We are using the cyanophage to disturb the cyanobacteria blooms prior to during the early stages of cyanobacterial bloom formation. It means we are just applying these cyanophages to kill the cyanobacteria, to kill the cyanobacterial blooms. Why? Because the cyanophages have the ability of the bacteriophage or cyanophage to break down the host in the lytic cycle is being explored to target specific host to reduce their numbers. Okay, I can explain in the more other ways. See, before going to the cyanophage treatment, we have to know about the cyanophages here. In case of the cyanophage classification, currently there are 3 families, because cyanophage classification up to the family level here. Currently there are 3 families, which are Mycoviridae, Podoviridae, and Scipoviridae. These 3 families came based on the morphology of the cyanophages. I don't want to go more explanation about the morphology and others. I'm just going for the cyanophage life cycle. What is the interlinking between the cyanophage life cycle and bloom control here? I already told cyanophage, we can use the cyanophage to kill the cyanobacteria or cyanobacterial blooms. Is it possible? Why it is really possible to use the cyanophores to remove the blooms in the water? Here, the cyanophores have its own life cycle to generate them itself, but we are using that advantage of life cycle of the cyanophores to remove the cyanobacterial blooms here. That is the only thing here. We are not doing something to the cyanophores to remove the cyanobacterial bloom here. The only thing is that it has an advantage we are utilizing of the cyanophages. What is that advantage means? The life cycle of the cyanophage, because cyanophages get involved in the two types of the life cycle. One is lytic cycle and another one is lysogenic cycle. Now here I am explaining only lytic cycle of the cyanophages, because cyanophages generally infect the cyanobacteria to reproduce, to replicate, to replicate their generations. They utilize the cyanobacteria to reproduce them. So here using that one to remove the cyanobacteria in the water system. Let's see the lytic cycle of cyanophage. Cyanophage infect the cyanobacteria and inject its DNA into the cyanobacteria. After injecting the DNA, it can overtake the cyanobacterial DNA and its replication mechanisms, and that DNA replicates itself in the host and produce the cyanophages and break the cyanobacteria and releasing into the water bodies. This is general concept of the life cycle of cyanophage. Now here we are taking that advantage of the life cycle of the cyanophage to remove the cyanobacteria. So there are few stages in the life cycle of the cyanophages. You can see here. The first one is penetration and replication and assembly and cell lysis. This is overall life cycle of the lytic phages. If you can see the right side figure, we can we can come to one stage like it is a graph that is called one-step multiplication curve of the phase. Here you can see all the steps in the one graph here. First one is penetration. As I told, the first phase can infect on the cell and release that DNA into the host cell. And the host, after inserting that DNA into the host cell and it started to the replication, that period we can call is a latent period. After making their generation, it goes for the burst cycle. Burst cycle means it can lyse the cell of the host. After making this, cyanophages can release into the water. This is the overall the cycle. Based on this mechanism, we are utilizing this type of the cyanophages to remove the cyanobacterial bloom, because cyanobacterial bloom, they have its own level to generate at a particular seasons. During that seasons, if we can able to release or introduce this type of the phages into the water, we can remove that type of the cyanobacterial bloom formation. This is our main motto. Host range of the cyanophages. Few cyanophages can infect only one cell, and some cyanophages can infect different type of the cyanobacteria. It means single type of phages can infect only one type of the species of cyanobacteria. It means species specific range of cyanophages. Some cyanophages, I mean one type of cyanophage can infect different type of the cyanobacteria. So overall we can say monovalent and polyvalent cyanophages. Monovalent cyanophages means capacity to infect only a single specific strain. Polyvalent means able to absorb bacterial surfaces, concern the ability to infect across the different bacterial species. So factors influencing the cyanophage and cyanobacteria interactions. There are a lot of factors which are influencing the cyanophage and cyanobacteria interaction. If the cyanophage cyanobacterial interaction is not good enough, the treatment efficiency will be decreased, because our aim is to make the good interaction between the cyanophage and cyanobacteria. Because of the interaction is not good, our treatment efficiency, I mean treatment efficiency means nothing but phage therapy is very less. So to enhance the interaction between the cyanophage and the cyanobacteria, we have to provide a good conditions. So we can say that yes, there are some condition, environmental factors, they can affect the cyanobacteria and phage treatment interactions, so that we have to provide a good environmental condition to make a better treatment efficiency. So until now, we discussed about the cyanophages and their interactions, how they are going to work and to remove the cyanobacterial blooms. But this slide showing the cyanophages in sustainable development. Is it really sustainable development by using the cyanophages to remove the cyanobacterial blooms from the aquatic systems? We have to consider before that. Two things we have to consider here. First one is disadvantages and second one is advantages. Certainly there should be a disadvantages. Of course, there should be a disadvantages. Whether it is possible to overcome from the disadvantages, we have to see, because we have to look upon the sustainable development. Sustainable developments, as we know, we have, it should be like regenerate. It is easy to handle and it should be like always available, never end process. So if you can go for the disadvantages, cyanophage infection is good to remove the cyanobacterial blooms. But the problem is cyanobacteria not only it produces the biomasses, it can also produce the cyanotoxins in the body. Once cyanophage infect the cyanobacteria, it will kill the cyanobacteria and it will break the cyanobacterial cell wall. Automatically cyanotoxins also will be released into the water bodies. That is the disadvantages nowadays. And what is the solution for that? Yes, there is some solution, because after breaking the cell wall, cyanotoxins are releasing into the water, but we have to consider the another treatment that is called flocculation. We can use some of the chemicals like aluminum related compounds which can accumulate and precipitate and get into down of the water bodies. During that time, there are another bacteria that can also degrade the type of the cyanotoxins. So that is one way of solution for these disadvantages. But in case of water treatment plants, this is the very, it is very negative impact on the water treatment plants, because in the water treatment plants there is no any other organisms to degrade this type of the compounds. In case of open systems like aquatic systems, there are a lot of other organisms. They can also degrade this type of the compounds after breaking the cells and coming the cyanotoxins into the water bodies. That is the one thing here. And second one is life cycle of these cyanophages are very long here compared to the bacteriophages, because bacterial life cycle is very less, just 20 minutes for the E. coli. In case of the cyanophages, it's like 2 days, 4 days like that for the each generation. So it is very long time to kill the cyanobacteria by using cyanophages. And the third disadvantages is tolerance of the host. After 1 or 2 generations, after treating with this type of cyanophages, the host getting the tolerance. After 4 or 3 generations, they are getting tolerance. They are preventing the cyanophage treatment. This is the one type of the disadvantages. So these are the main 3 disadvantages. But what are the advantages of cyanophage treatment? Cyanophage treatment, if you go for the cyanophage treatment, if you go for the cyanophage infection to the any cyanobacteria, few reports are very clearly indicating that the cyanobacteria which is able to produce the cyanotoxins are preventing after treatment with the cyanophages. See the wonder here. Few reports already very clear that cyanophage infection was able to found shift the host community from toxin producing to non-toxin producing population. It means after infection with cyanophage, the production of cyanotoxins are preventing. It means it preventing the production of the cyanotoxins. That is the great advantage after treatment with the cyanophages. And second advantage is cyanophage infection was also found to the same thing. And second one is each cyanophage its own stick to host range. See, in case of the chemical treatment, that chemical can also shows effect on the another organisms, non-target organisms. In case of cyanophage treatment, it cannot infect others. It means no negative impact on non-target species. This is the advantage of the cyanophages. Apart from that, recently researchers are focused on synthesizing the artificial cyanophages to enhance the interaction between the cyanophage and their hosts. Those include modifying the recognizing receptors, because if you want to enhance the cyanophage treatment, cyanophage should absorb on the cyanobacterial cells. To absorb more cyanophages, there should be a more treatment efficiency. To enhance the absorption's ability, enhancing the lysogeny and excluding the defense of the hostess. If we can go for the artificial cyanophages, if we can bring changes in the cyanophages, if we can modify the genetic level, you can go for the many changes, like you can go for the absorption ability, enhancing the absorption ability, enhancing the recognizing receptors, enhancing the lysogeny, and enhancing and excluding the defense of the host. So these are the advantages if we succeed in making the artificial cyanophages. So in the research studies in Korea, currently we are focusing on the lakes directly. We are focusing on the
Excuse me, sir.
yes, please.
So actually, our next session will be
Okay.
come.
Yeah, it took a long time, right? Anyhow,
Yeah.
I am stopping now here. Okay. And okay, sir, thank you for giving this opportunity to give a great platform internationally. And thank you very much.
Thank you so much, sir, for your insightful lecture on phase therapy for the sustainable management of cyanobacterial harmful algal blooms. Your presentation was both informative and valuable. Despite your busy schedule, your dedication to research was evident as you took the time to share your knowledge with us. Thank you so much, sir.
Thank you very much, sir. Thank you.
Actually, the time is for break, but the participants number are very high. That's why we are not take a break. Now our oral and poster presentation session will be start. And the first participant is Suraj, then Megha, Anjali Rajput, Kanchan, and Dr. Suman. So first participant is Mr. Suraj.
Yes, sir.
Yes, please present your paper.
Okay.
Please, every participant should maintain the time limit. The time limit is 5 to 7 minutes.
Is my presentation is visible to everyone?
Yes.
Okay, am I audible?
Yes, very much audible.
Okay. So first, thanks for giving me this opportunity to present my thoughts or ideology to this international conference. Okay. Effect of aloe vera leaf extract on normal skin cancer. ओके, जैसे हम जानते हैं कि हमारे स्किन पे बहुत सारे कमेंसल्स हैं, वो माइक्रो एनवायरनमेंट के अकॉर्डिंग रहते हैं। ओके, स्किन के डिफरेंट लोकेशन पर डिफरेंट टाइप के माइक्रो ऑर्गेनिजम्स हो सकते हैं। ओके, स्टेफाइलोकोकस एपिडर्मिडिस लाइक स्किन के सरफेस पे प्रेजेंट रहता है। प्रोपियोनी बैक्टीरियम एक्ने, ये हेयर फॉलिकल में प्रेजेंट रहता है। ओके, तो अकॉर्डिंग टू द माइक्रो एनवायरनमेंट हमारी स्किन किस एरिया में किस माइक्रो एनवायरनमेंट को क्रिएट कर रही है, उस अकॉर्डिंग डिफरेंट टाइप के जो माइक्रो ऑर्गेनिज्म है वो डिफरेंट टाइप के जो माइक्रो ऑर्गेनिजम्स हैं वो हमारे स्किन पे प्रेजेंट है। ओके, तो कुछ चीज़ें ऐसी हैं जो हम इस डेली लाइफ में अपने डेली रूटीन में करते हैं जो इस माइक्रो ऑर्गेनिज्म के जो माइक्रो इन्वायरमेंट है उनको डिस्टर्ब कर सकती है। लाइक इफ any cosmetics we are applying on our skin that can may change this micro environment. Okay, and any change in this micro environment lead to the disease formation. Okay, because all the commensals that are present over on our skin can cause disease or can act as a opportunistic pathogen. Okay, so aloe vera gel, जैसे कि हम जानते हैं कि एलोवेरा जेल बहुत ही कॉमनली यूज्ड नेचुरल प्रोडक्ट है। इसे कॉस्मेटिक्स बनाने में भी यूज किया जाता है इंडस्ट्रीज के द्वारा, और हम नेचुरली भी इसे 96% ऑफ वाटर से 4% इसमें ड्राई मास रहता है। इस ड्राई मास में 6.86% प्रोटीन रहता है, फैट 2% के अप्रोक्स रहता है, कुछ फाइबर्स रहते हैं 73% और कुछ विटामिन सी रहता है, एस्कॉर्बिक एसिड एंड कुछ एश रहते हैं। ग्लूकोमेनन एंड हेमिसेलुलोज फाइबर्स प्रेजेंट रहते हैं जो स्किन के टेक्सचर के लिए बहुत ज्यादा इंपॉर्टेंट रहते हैं। हाइलुरोनिक एसिड, हेपेरिन, एक्मेनिन और कुछ म्यूकोपॉलीसैकेराइड्स रहते हैं इसमें। उसके अलावा कुछ एंजाइम्स ये कंटेन करता है, एल्केलाइन फॉस्फेटेज, अमाइलेज, कैटालेज, लाइपेज, प्रोटीएज, क्रिएटिनेज एक्सेट्रा। कुछ फिजियोलॉजिकल फंक्शनिंग रहती है एलोवेरा की जो हमारे स्किन के लिए हेल्पफुल भी हो सकती है, हमारे स्किन के अगेंस्ट भी जा सकती है। कुछ पॉलीसैकेराइड्स जो एलो जेल में प्रेजेंट हैं वो हीलिंग का काम करते हैं, एंटी फंगल एक्टिविटी शो करते हैं, एंटी डायबिटिक इफेक्ट रहता है, एंटी इन्फ्लामेटरी, एंटी कैंसर, इम्यूनोमोड्यूलेटरी एंड कुछ गैस्ट्रो प्रोटेक्टिव प्रॉपर्टीज रहती हैं एलोवेरा में अगर इसको हम एज अ सप्लीमेंट भी यूज कर रहे हैं तो। उसके अलावा इंटेस्टाइनल एब्जॉर्प्शन जो है वो काफी हद तक इम्प्रूव होती है एलोवेरा सप्लीमेंट लेने से। इसके अलावा ग्लूकोमेनन हेमिसेलुलोज जो फाइबर्स हैं वो हीलिंग में काफी ज्यादा पार्टिसिपेट करते हैं। अगर कहीं पे घाव है स्किन पे उसको बहुत जल्दी हील कर सकते हैं। हमारे जो सेल्स हैं उनकी प्रोलिफरेशन को बहुत ज्यादा बढ़ा देते हैं। जो नॉर्मल फ्लो है ब्लड का, लिम्फ का, गैसेस एक्सचेंज का, रेस्पिरेटरी ट्रैक जो एल्वेलाई है एंड पे, उसमें जो गैसेस का फ्लो है उनको इंक्रीज किया जाता है। उसके अलावा है एलोवेरा, राइट। अब साइड इफेक्ट्स में क्या है कि जो हाइड्रोजेंथिया थ्रेसिन है, ठीक है, उससे ड्राइव्ड जो भी नेचुरली एलो है वो स्ट्रॉन्ग परगेटिव एक्शन शो करते हैं और ये कहीं ना कहीं ह्यूमन के लिए एक थ्रेट भी है, ठीक है। लॉन्ग यूज ऑफ एंथ्राक्नोन में रिजल्ट इन द इंटेस्टाइन इन्फ्लामेशन, इंटेस्टाइन में इन्फ्लामेशन कॉज करता है लॉन्ग टर्म यूज इसका। एंड जो प्रोलॉन्ग यूज है वो मेंस्ट्रुअल ब्लीडिंग में भी डिफिकल्टी देखने को हमें मिल सकती है। एक्सेसिव कंजप्शन ऑफ एलोइन थ्रू एलोवेरा may not only a strong laxative effect but also toxic effect. Apart from that, एलोइन इनहिबिट लिपिड पर ऑक्सीडेशन, जो लिपिड का मेटाबॉलिज्म है लिपिड पर ऑक्सीडेशन से उसको भी कहीं ना कहीं बहुत हाई इफेक्ट करता है। और कुछ एलोवेरा की स्पीसीज ऐसी हैं जो काफी टॉक्सिक रहती हैं, वो टॉक्सिन रिलीज करती हैं, उनका कंजप्शन बिल्कुल अवॉइड करना चाहिए। कुछ लाइक कुछ फॉरेस्ट ऐसे हैं जहाँ पे स्पीसीज पाई जाती हैं। कुछ स्किन सरफेस और हेयर फॉलिकल के कुछ कॉमन कमेंसल हैं, इसमें कुछ फंगस इंक्लूडेड है और कुछ बैक्टीरिया है। बेसिकली अपना न्यूट्रिशन लेते हैं स्किन की सिक्रीशन से, लाइक स्वेट है, सेबम है, कुछ और सेल जनरली क्या होता है, ये जो कमेंसल है ये हमारे लिए पैथोजन नहीं होते, बट ये हम इम्यूनाइजेशन का भी काम करते हैं। हमारी स्किन पे रह के ये कुछ इम्यूनाइजेशन का काम करते हैं, जो दूसरे माइक्रोबियल डिसीज हैं उनसे हमें प्रिवेंशन दे सकती है। Commensals generally contribute effectively in our immune system. The skin property is different from one region to the body part of another, as does the composition of skin's microbiota. The availability of nutrition and moisture partially dictate which microorganism will thrive in the particular region of skin. इस पॉइंट में यही डिस्कस किया गया है कि हमारे स्किन का जो माइक्रो इन्वायरमेंट है वो डिफरेंट लोकेशन पे डिफरेंट है। हमारे हेड में अलग होगा, लाइक हैंड्स में अलग होगा। दे आर जनरली एरोबिक, एनारोबिक एंड फैकल्टेटिव एनारोब्स। अब जो माइक्रो इन्वायरमेंट है उसके अकॉर्डिंग जो स्किन कमेंसल है वो अलग अलग मिलेंगे हमें। अब जैसे कॉर्नेबैक्टीरियम एक्ने है, वो एक एनारोबिक बैक्टीरिया है। क्यों? क्योंकि वो हेयर फॉलिकल्स में, लाइक हेयर फॉलिकल की लोकेशन पे हमें मिलता है। वैसे ही जो स्टेफाइलोकोकस एपिडर्मिडिस है वो एक एरोबिक बैक्टीरिया है, वो फ्री रहता है स्किन सरफेस पे। उसके अलावा कुछ फैक्टर्स हैं जिनके डाइवर्ट होने पे या ऑल्टर हो जाने पे हमारे नॉर्मल स्किन कमेंसल एक अपॉर्चुनिस्टिक पैथोजन की तरह बिहेव करने लगते हैं। अगर पीएच चेंज हो रहा है स्किन का, जो कि एलो से लगभग होता ही है, हमारे जो स्किन का नॉर्मल पीएच है वो आउटर एनवायरनमेंट पे डिपेंड करता है। आउटर एनवायरनमेंट कैसा है और जो लिम्फेटिक फ्लूइड है वो किस टिश्यू में कैसा है। बट अगर हम एलो यूज करते हैं डायरेक्टली एज अ नेचुरल प्रोडक्ट, तो 1 पीएच तक लो कर सकता है स्किन का पीएच, बिकॉज एलो का जो नेचुरल नॉर्मल पीएच है वो 4 to 5 के बीच में रहता है। इसके अलावा अगर टेम्परेचर चेंज होता है, ओके, उसकी वजह से हमारे स्किन कमेंसल अपॉर्चुनिस्टिक पैथोजन की तरह बिहेव कर सकते हैं। इसके अलावा जो नेचुरल न्यूट्रिशन स्किन उसे दे रही है हमारे माइक्रोबायोटा को, तो अगर वो किसी रीजन से चेंज होता है, जो कि एलो यूज करने से ऑब्वियसली चेंज हो सकता है, हमारी बॉडी के सिक्रीशंस एलो पेस्ट यूज करने से या डायरेक्ट एलोवेरा यूज करने से चेंज हो सकती है। वाटर एक्टिविटी, ड्राइनेस ऑफ द स्किन, दैट मीन्स अगर स्किन ड्राई हो या बहुत ज्यादा वैक्स स्किन पे आ जाए, वैक्स की तरह ही लाइक एलो जेल है वो वर्क करता है, ओके। उसके अलावा जो ऑस्मोटिक प्रेशर है वो भी एक रोल प्ले करता है। कैन एलोवेरा इनहिबिट द ग्रोथ ऑफ स्किन कमेंसल? ये एक अपने आप में ये क्वेश्चन है, क्योंकि कुछ लाइक प्रूफ या कुछ एक्सपेरिमेंट्स ने प्रूव किया है कि जो एक्मेनन है, लाइक ये कंपोजिशन है एलोवेरा की, ये किसी किसी केस में बैक्टीरियोसाइडल प्रॉपर्टी भी रखती है, वायरोसाइडल होती है, फंजीसाइडल प्रॉपर्टीज भी कैरी करती है। जिसका सीधा सीधा मतलब ये रिप्रेजेंट होता है कि अगर हम एलो को यूज करते हैं तो कहीं ना कहीं हमारे माइक्रोबायोटा को लाइक वैनिश करने में, हमारे माइक्रोबायोटा के नॉर्मल माइक्रो एनवायरनमेंट को ये डिस्टर्ब करती है। इसके अलावा कुछ एलोवेरा एंटीऑक्सीडेंट प्रॉपर्टीज कैरी करते हैं, साइटोटॉक्सिक इफेक्ट शो करते हैं हमारे स्क्वैमस सेल पे, ओके। और ये क्या करता है, उस लोकेशन पे जो भी हमारा माइक्रोबायोटा प्रेजेंट है वहां पे बैक्टीरियोस्टैटिक इफेक्ट शो करता है, और मेनली ये देखने को मिलता है स्ट्रेप्टोकोकस के जेनेरा में। बैक्टीरियोस्टैटिक का यहाँ क्या मतलब है कि वो हमारे माइक्रोबायोटा को किल तो नहीं करेगा, बट माइक्रोबायोटा की जो ग्रोथ है उसको बिल्कुल ही स्टॉप कर देगा। अब यहाँ पे मेनली है कि एलोवेरा का मोस्टली यूज होता किस लिए है। एक तो हम नेचुरली एलोवेरा का यूज करते हैं एज अ पेस्ट अपनी स्किन पे, उसके अलावा एज अ सप्लीमेंट हम यूज करते हैं, और इसका जो डायरेक्ट कंजप्शन है वो एंटीऑक्सीडेंट प्रॉपर्टीज शो करता है, हमारे सेल प्रोलिफरेशन को इंक्रीज करता है। इसके अलावा कुछ इंडस्ट्रीज हैं जो एलोवेरा बेस्ड प्रोडक्ट बनाती है, लाइक एक फेमस कंपनी है फॉरएवर लिविंग कंपनी, वैसे तो एक नेटवर्क मार्केटिंग कंपनी है, बट वो एलोवेरा कंपनी एलोवेरा बेस्ड प्रोडक्ट बनाती है अपने सारे। उसके अलावा अब कंक्लूजन से पहले मैं ये कहना चाहूंगा कि हमने दो चीजें देखी, एलोवेरा के इफेक्ट देख लिए हमने कि वो क्या साइड इफेक्ट शो कर रहा है, वो बैक्टीरियोसाइडल प्रॉपर्टीज शो कर रहा है, और दूसरा हमने ये देख लिया कि एलोवेरा में कुछ ऐसी चीजें हैं लाइक मॉइस्चर है, वो स्किन के टेक्सचर को अच्छा रखता है, ठीक है। तो यहाँ पे सवाल ये आता है, क्या नेचुरली या विदाउट किसी साइंटिफिक स्टडी के एलोवेरा को अपने स्किन पे यूज करना और उसके लिए उसके कॉस्मेटिक्स बनाना, स्किन के टेक्सचर को इम्प्रूव करने के लिए, क्या ये लाइक हेल्पफुल है हमारे माइक्रोबायोटा को भी नॉर्मली उनकी ग्रोथ के लिए, या फिर ये किसी ना किसी तरह हमारे माइक्रोबायोटा को लाइक वैनिश करेगा, हमारे इम्यूनाइजेशन को इफेक्ट करेगा, और एक लाइक अपॉर्चुनिस्टिक पैथोजन भी उन्हें बना सकता है। तो इसी चीज पे मेरा ये थॉट है, ये मेरी आइडियोलॉजी है कि मुझे इस पे थोड़ा काम करना है और ज्यादा। थैंक यू।
एलोवेरा में ऐसा कौन से कंपोनेंट्स होते हैं जिसकी वजह से स्किन मतलब सही रहता है?
एक्चुअली स्किन के सही रखने में जो मोस्टली काम है वो वाटर कंटेंट का ही है, ये हाइड्रेट रखता है।
नहीं, उसमें कोई
उसके अलावा
कंपोनेंट्स
जो
होते हैं
एलोवेरा,
स्पेशल?
जी
उसमें कोई स्पेशल कंपोनेंट्स होते हैं
स्पेशल कंपोनेंट्स
जो स्किन के लिए
फॉर द स्किन
यूजफुल है इन
टेक्सचर,
एलोवेरा?
फॉर स्किन टेक्सचर यू आर सेइंग?
यस।
एक्चुअली देखो, जैसे मैंने बताया था ग्लूकोमेनन है जो, ठीक है, वो वाउंड हीलिंग में काम करता है। तो सेम ग्लूकोमेनन अगर थोड़े से अमाउंट में हो तो वो प्रोलिफरेशन को इंक्रीज करता है स्किन सेल्स की। तो मेरी नॉलेज में सिर्फ यही एक ऐसा लाइक कॉम्पोजिशन है उसका जो काफी हद तक टेक्सचर को मेंटेन कर सकता है स्किन की। इसके अलावा वाटर कंटेंट का ही मेजर रोल होता है, हम वाटर कंटेंट के लिए ही यूज करते हैं एलोवेरा जेल को।
ओके ओके। नेक्स्ट मेधा। नेक्स्ट मेधा।
Yes, sir.
यस, प्लीज प्रेजेंट योर पेपर।
Yes. So, I
You are not audible.
hello, sir. Now, sir, am I?
Yes,
Hello.
please present your paper.
So, actually, some network problems. So sorry for that. Sir, is my presentation is visible?
No.
Okay, sir.
You may take time.
Now, sir.
Yes, now visible.
Good afternoon. Okay, sir. Thank you. Good afternoon, everyone. Today's my topic is and my idea is basidomycetes, how they are helpful for human welfare. As we know that basidomycetes are useful in the degradation of the cellulose in the plant. The algae, which is helpful mainly in degradation of the plant cellulose, is mainly white rot and brown rot fungi. Algae. Basidomycete is the one of the class of the fungi that reproduce sexually with the help of basidospores and are generally sporophytic in nature. They are used in the wide industry aspect for fermentation and the antibiotics because there is their secondary metabolism and exogenous enzymatic action on the organic matter like degradation of lignin and the cellulose in the plant biomass. They play a very economical role in the waste management of organic distribution. Like we see that in a forest or in anywhere, they degrade the waste woods cellulose which are and lignin which are present in the wood by producing the endo enzymes. Now the role, I would like to explain the enzymes which are present in basidiomycetes and they what they do and what's their function of the degradation of the cellulose and lignin. First enzyme is endoglucanase. Second one is cellulose bihydrolase, beta galactoside, lignin peroxidase, magnesium peroxidase, versatile peroxidase, and phenol oxidase. Endoglucanase. Endoglucanase are enzyme that breaks down cellulose. They are used in the plum and paper industries and in biofires. Endoglucanase actively is important for industry and for studying cellulose degradation. Endoglucanases have the following characteristic: enzyme classification of the glucanases. Its activity is glucanase randomly cleaves the cellulose polymerase into small sugar and oligomeric polysaccharides. And its industrial uses, gluco, uh, glucogena- glucanases are used to degrade cellulosic complex and plant biomass. Cellulose degradation, they do by cleaving the internal beta galactosidic bond and in the cellulose chain. Biodegradation is endoglucanase convert linoscellulosic substance to soluble sugar. The second enzyme is biohydrolase. It is known as CBH enzyme that breaks the cellulose and release the sugar molecules and CBHR is a part of the glycosylic hydrolase family. They have tunnel like activity side of pros, cleaving the cellular biomass units from the end of the cellulose chain. Now we will what they do is they actively perform in 50 degrees Celsius maximum optimum temperature for them for there to perform the ac- catalytic activity. Now the second enzyme is beta glucosidase. Beta glucosidase is an enzyme that catalyze and the hydrolysis of the glycosidic bond in a cellular bios. The reaction produce glucose, which is important step for the effective utilization of cellulose. Beta glucosidase is a key component of the cellulose in the system enzyme metabolic cellulose. So basically, beta cellulose, what they do, they hydrolyze the terminal non-reducing beta and D glucosidase reduce of a gluco glucosidase, then hydrolyze the oligosaccharide to the glucose. The second enzyme is lignin peroxidase. Lignin peroxidase is a fungal enzyme that plays a key role in the lignolytic cycle. The lignolytic cycle is the structural component of the plant walls is degraded. When the plant's cell wall is degraded, that is done by the lignin peroxidase. And the next enzyme is versatile
हाँ वो लेट चल रहा है शेड्यूल
peroxidase.
थोड़ा सा, तो आपको 2:30 का अराउंड होंगे। जी जी।
Versatile peroxidase enzyme. This is also helpful in the degradation of lignin. They produce the typical lignin peroxide, then they perform the catalytic activity which helps in the breakdown of the lignin. So basically these enzyme are uh, this enzyme are work collectively or they present in one uh, singly in the environment, and they do the job of degradation. So like we know that the I would like to conclude we know that the older age peoples face the digestive problem of the cellulose. They are the cellulose deficiency for the degradation of it. So why we would use it as a probiotic and provided it to the manufacture it in a industrial way and give it to the people who are depressed of the degradation of the cellulose, compromised people. Thank you, sir.
Okay, thank you. Next participant, Anjali Rajput. Next, Anjali Rajput. Next, Kanchan. Next, Kanchan.
Yes, sir.
Yes, please present your paper.
Sir, is it visible?
No.
So now it is visible.
Yes, yes.
Okay. Good afternoon, everyone. My name is Kanchan Sangole. I am an assistant professor, Department of Botany, Nabila Mahavidyalaya, Katur. Today's title of my poster presentation is QR code generation in Botanical Garden, Bajaj College of Science, Varta. The abstract is QR code stand for Quick Response Code, which is trademark for the type of matrix barcode which was invented by the Japanese corporation Denso Wave. QR code has a number of features such as large capacity of data encoding, high speed reading, small printout size, 360-degree reading, structural flexibility of application, and quick availability of data. The present study deals with advantages of evolving innovation trends and taxonomy over the conventional techniques. The keywords are QR code, plants morphology, scientific names, and plant medicinal uses. The goals are to encourage engagement with the garden and its exhibit by providing most interesting experience to the visitors, to study morphological characters and plants belonging to the various families, and to study the medicinal and aesthetic values of plants. The method of making QR code is first identify the plant with the help of PlantNet and Floras. Then add all characters of plants, that means botanical name, local name, morphology, medicinal uses and aesthetic value on the Word file. After that, convert Word file into JPG image. Then record audio with the help of sound recorder. Enhance the audio clips with the help of Adobe Podcast. Make video on ClipJam with the addition of JPG image and recordings. Then upload video on YouTube, copy video link from YouTube and paste it into Adobe Express and generate QR. In the result, we got a QR code. In this research work, I work on total 23 plants. Some are listed here, that is 20 plants are listed here. Please scan QR and get additional information about plants. And some references are here. Thank you.
Okay. Thank you, ma'am. Next participant, Anjali Rajput. Anjali Rajput.
Good afternoon, everyone. Am I audible?
Yes, very much audible.
Good afternoon, everyone. I'm Anjali Rajput, PhD scholar and Professor Veena Sharma from Ministry Vidyapeeth. I appreciate the opportunity to share insights on my topic, lead toxicity. And Tanya, can you please present my presentation? Hello?
Anjali, wait.
Okay.
So is it visible?
Yes, Tanya.
Yes, visible, very much visible.
Okay.
Anjali, start.
So, yeah. I appreciate the opportunity to share insights on the topic lead toxicity and cardiovascular consequences, insights into oxidative stress, and other intricate pathways. Tanya, please change the next slide. Okay, so the insights into this presentation topic will be the introduction that how lead affects the cardiovascular system. The main cause of lead toxicity is basically oxidative stress generation, but apart from it, lead is also involved in various other mechanisms that lead to the cardiovascular consequences. So starting with the introduction topic, lead exposure today remains a pressing concern in the realm of public health, with a mounting body of evidence underscoring its adverse impact on cardiovascular well-being. This exposure to lead instigates the production of reactive oxygen species, leading to the consequential cellular and physiological damage. So, lead is basically a highly hazardous and prevalent environmental pollutant due to its ability to cause biochemical and physiological changes. So, basically, ongoing research and environmental stewardship are crucial to protect the global health and ecosystem. Therefore, the significance of this presentation lies in the fact that it will help us to gain some insights on how to save ourselves from the lead. Tanya, please change the slide. Okay. So, the molecular mechanism of lead-induced toxicity is associated with the initiation of oxidative stress by elevating the levels of reactive oxygen species such as superoxide radicals, hydrogen peroxide, hydroxyl radicals. Basically, uh, ROS are generally produced either by mitochondrial electron transport chain present in nearly every cell type. While ROS production is essential for maintaining homeostasis, regulating cell signaling, supporting immunity, metabolic control, but the diverse pathophysiological conditions can intensify the ROS production, compromising the capacity of the antioxidant defense system. So, this further leads to oxidative stress, which is characterized by an imbalance between the production of free radicals and the biological system's ability to counteract or repair the damage. So, the resulting ROS accumulation can lead to inflammation, fibrosis, apoptosis, and cellular death, underscoring the multifaceted impact of lead exposure on the biological system. So, lead impacts various antioxidant enzymes as well, such as SOD, GSH, GST, thereby decreasing the immunity of the body. So there are certain reactive oxygen species that you can see: superoxide radicals, hydroxyl radicals, hydrogen peroxide, and singlet oxygen. Superoxide basically reacts with the iron sulfur proteins, and hydroxyl radical is extremely reactive with the DNA. It can generate the cuts within the DNA, leading to the epigenetic perturbations. And then we have hydrogen peroxide that reacts with proteins, heme proteins, and DNA, thereby affecting the basic cellular structure. Tanya, please change the slide. So apart from reactive oxygen species, there are other intricate mechanisms as well that can lead to the lead toxicity. So basically, regardless of recent progressive advances in the treatment of cardiovascular diseases, they remain the leading cause of mortality and a substantial contributor to the global disease burden. Factors such as environmental toxicants, including lead and other heavy metals, can actually lead to the cardiovascular outcomes, including stroke, coronary artery disease, peripheral arterial disease, hypertension, and beyond oxidative stress, the cardiotoxicity induced by lead engages diverse mechanisms, including endothelial dysfunction, inflammation, and dysregulation of the renin angiotensin aldosterone system and the disruption of calcium homeostasis. So let's talk about our first mechanism now. Tanya, please change the slide. Okay, so coming to the inflammation and endothelial part, basically what happens, lead exposure triggers an inflammatory response within the cardiovascular system. It triggers the activation of endothelial cells that line the inner surface of blood vessels. So these activated endothelial cells release pro-inflammatory cytokines such as interleukin 6 and tumor necrosis factor alpha, along with some adhesion molecules as well. These recruited immune cells release inflammatory mediators that further leads to the chronic inflammation causing tissue damage, fibrosis, and onset of atherosclerosis and cardiac disease progression. Also, when we talk about endothelial function, that disrupts the endothelin levels and sensitivity. It elevates the ET1 levels, which basically leads to the impaired vasodilation and increased vasoconstriction. So increased vasoconstriction will lead to the progression of cardiovascular pathologies like hypertension, thrombosis, atherosclerosis, etc. Taya, please change the slide. Tanya,
I have changed the slide, Anjali.
yeah, it's visible now. Thank you, Tanya.
1 minute.
Coming, yeah.
Yeah.
Now coming to the LAD and renin-angiotensin pathway, RAS basically plays a crucial role in regulating blood pressure, vascular tone, and the balance of salt and water in the body. This system comprises of three primary components, which is basically angiotensin, sorry, renin, which converts angiotensinogen into angiotensin, and then you have ACE, which converts angiotensin 1 into angiotensin 2. Angiotensin 2 is basically a potent vasoconstrictor that stimulates the release of aldosterone, which encourages the sodium and water retention. But what happens? It affects the blood pressure and fluid balance, and this heightened lead actually leads to the heightened activity of ACE, which leads to elevated intrarenal angiotensin 2, the primary mediator of physiological effects of RAS, including blood pressure regulation, volume regulation, and aldosterone secretion. The influence of lead exposure on the systemic renin angiotensin aldosterone system in experimental animals basically it depends on lead exposure, the dosage, but since angiotensin 2 is a vasoconstrictor, it leads to the it affects cardio it affects cardiac cells in a negative manner, leading to various outcomes like again hypertension, atherosclerosis, and myocardial infarction. Taya, please change the slide. Thank you. Okay, coming to the disruption of calcium homeostasis, which is basically the most important cause of lead toxicity. What happens? Lead disrupts calcium homeostasis in cardiac cells by interfering with the calcium signaling, uptake, and release. Lead exposure affects various calcium channels in cardiac cells, including L-type calcium channels and iodine receptors. These L-type calcium channels, these calcium channels facilitate the calcium influx during the action of potential, while ryanodine receptors participate in calcium release from the sarcoplasmic reticulum. Uh, lead actually leads to the dysregulated calcium levels resulting from the elevated intracellular calcium, which directly affects the contractility of the this vascular smooth muscle cells by restraining the activity of sodium potassium pump, leading to the altered myocardial contractility and vascular resistance. Um, Tanya, please change the slide. Okay, coming to the prevention and treatment. The lead-induced cardiac toxicity is a concerning health issue, and recent scientific research has shed light on effective prevention and treatment strategies. So, therefore, understanding the mechanism of lead's detrimental effects on the heart has paved the way for targeted interventions to protect those at risk. So, efforts are required to mitigate lead exposure and its cardiovascular consequences, which are of utmost importance. For example, the development of reliable biomarkers for lead-induced cardiac toxicity can facilitate the early detection and intervention. These biomarkers can actually offer us the potential in identifying the individuals at higher risk and monitor their cardiac health amid lead exposure. Ultimately, there exists pressing necessity to formulate and execute regulatory and public health interventions directed at the prevention and reduction of lead exposure levels, particularly in vulnerable population. So these endeavors should surpass existing intervention measures to guarantee the sufficient protection of public health. Tanya, please change the slide. Tanya.
Anjali, I have changed the slide.
Okay, thank you. So, concluding this presentation with the fact that lead-induced cardiac toxicity poses a notable health risk because of its detrimental impact on the cardiovascular system. So, scientific investigations are required, and also there are few investigations that has shown that lead exposure initiates oxidative stress, disrupts vascular function, and interferes with the calcium homeostasis. So these factors collectively contribute to the initiation and progression of conditions such as atherosclerosis, hypertension, along with altered cardiac function and structural myocardial damage. So it is important to acknowledge that lead-induced cardiotoxicity is a multifaceted phenomena involving interdependent mechanisms. So basically, by collaborating at various levels, we can reduce the burden of lead-related cardiovascular complications and improve the overall cardiovascular health of affected individuals and communities. Thank you.
Thank you so much. Now the next participant.
Thank you, sir.
Dr. Suman. Next participant, Dr. Suman. Next participant, Aishwarya. Next participant, Aishwarya. Aishwarya Sharma.
Hello, sir. This is Dr. Suman. Am I audible?
Yes, ma'am. Yes, ma'am. Audible.
Okay. Thank you.
Thank you.
Good afternoon, everyone. And first of all, thank you, organizing team, for giving me the opportunity. My presentation is visible.
No, no, not ma'am. No visible. Not visible.
Please give me a minute.
Ma'am, your PPT is not visible.
I'm trying.
Yeah.
Now,
Yes, ma'am. Yes, ma'am.
is it
Visible.
okay? Thank you. Topic of my presentation is flower petal extract of Taget petula as an insecticide on tomato plants. As we all know that marigold is a popularly grown flowering plant. Taget species is a plant of various uses, having ornamental, medicinal, food, and forage applications. Also, the bloom of French marigold are frequently employed, particularly for treatment of diseases associated with inflammation. It is also known as a medicinal herb, which is used to treat a variety of illness and has great therapeutic effect. This marigold is having phenolic compounds and terpenes, and beside this, it is also having monoterpenes, hydrocarbons, and monoterpene ketones, and all these are having like various properties, like they are having insecticidal, larvicidal, mosquitocidal properties. Also, some target species have a substantial impact on the nematodes also. After reviewing the literature, we found the lacunae, and the aim of our study was to develop a biopesticide that will be economically acceptable to the Aligarh climate. The study was done on the district Aligarh of UP. The experiment was performed on tomato agriculture field of village Zalalpur, Khair Road, Aligarh district, and the other requirements were tomato plants, targets flower, and we identified the plots for planting the tomato plant. Then we also selected the checked the soil that will be suitable for their cultivation, watering can or sprayer, and other measuring instruments. And to perform our experiment, five sites were selected from 1 to 5, each site with an area of 10 by 10 as an experimental plot, and they were in the field only. One plot with natural control, and while other on which we use different concentration of marigold flower extract. The grasses and weed of the experimental plot were removed
Ma'am,
time to
please
time.
change your slide.
Sorry,
Please change the slide. It's not
I'm
moving.
changing here. Actually, I'm changing it my side, but I don't know why it is not visible to you. Hello?
Hello.
Sir, I'm changing it from my side.
Okay, but
I don't
here
know.
it's not visible.
I don't know. I'm at neck. I'm changing with my words. Uh,
Yeah, yeah. Now it's visible.
Now visible. Okay, thank you. Please confirm again. Is it visible now?
Yeah, yeah. It's visible now.
Okay. These are our five experimental. These were our five experimental site. Site one with control, with no extract, and the other four experimental site 2, 3, 4, and 5 with where we use the different concentration of the marigold flower extract. Can you see the next slide now?
Yes.
Okay, thank you. So how we prepared this aqueous marigold flower extract? For this, we used 50 gram of dried powdered marigold flower, and then were weighed and then mixed in 500 ml of distilled water. And the solution was boiled, cooled, and filtered through Wattman filter paper. And then this filtrate was kept at room temperature, and different concentration were made, like 20%, 40%, 60%, and 80%. And these concentration were sprayed on the tomato plants on a weekly basis from 10th day onwards. This is how we prepared this aqueous marigold extract in the lab. And the for data collection, we measured the growth, yield, and disease parameter. Here I'm showing only growth and yield of the tomato plants. Uh, Tomato plants were morphologically measured from 20 day to 120 days at an interval of 15 days after transplantation till harvest. The number and mortality rate of caterpillar was also recorded at different concentration of marigold extract. Then shoot height, number of leaves, number of bud, flowers, and number of fruits were also measured, which were taken as a growth factor. Uh, these are the figures showing the measurement of leaf and fruit after using the marigold flower extract. I hope slides are moving now. This
No,
slide is showing.
this is the experiment sites.
Oh, I don't know what is the problem. I'm changing from my side, but I don't know why the changes are not visible to all of you.
Okay, now the slide is result.
Okay, thank you.
Okay.
Yes. Is it changing now?
Yeah, now changing. It's a graph
Okay,
slide.
okay. Yes. This image is showing the caterpillar images with their mortality rate at different concentration. And similarly, we made the graph of which is showing the mortality of the various caterpillar at different concentration of marigold flower extract. And also, we found that increase in plant height in case of the experimental site as compared to the control where we use the flower extract. Similarly, number of leaf were also increased in the experimental sites, and number of branches as well as number of, we found an increase in number of birds and number of flowers, and number of fruits were also in significantly found to be increased in case of the experimental site as compared to the control group. Here we are showing a small part of our study, so we can summarize, I can summarize the result here. The mortality response over time show a steady dose dependence. It was interesting to note that the highest aqueous marigold flower extract content was linked to the highest mortality. The marigold flower extract significantly increased the number of branches, shoot height, quantity of tomato plant leaves, buds, blooms, and fruits, where significantly less tomato illness were present than in the control group. So we can conclude that the results show that all four experimental sites in tomato field had a significant difference in fruiting. Thus, this study highlights the efficiency of marigolds extract as a natural pesticide, and it also goes with an advantage of marigold-based insecticides for environment and their possible uses, with special emphasis on their function in integrated pest management plans. Slides are moving now.
Yes, moving.
Oh, thank you. These are our MSc students who performed the whole experiment. I'm just presenting their results. Thank you.
Okay. Thank you so much, ma'am.
Thank you.
Now next participant is Surja. Next participant, Surja. Next, Bikram. Next participant, Bikram. Next participant Vikram. Next Monali. Next Monali. Nikesh. Yasika. Bansikha. Anyone here? Next participant Akanksha Burma from BVAU Lucknow. Next participant Akanksha Burma BVAU Lucknow. Akanksha Kumari.
Nancy.
Akanksha Burma, please proceed. Akanksha Burma, you are not audible. Next participant Ankit Kumar. Next participant Ankit Kumar. Biplavi. Bikram Singh. Mr. Bikram Singh.
Good afternoon, everyone. I'm Akansha Rathore, currently pursuing my MSc in Zoology from Baba Sahib Bhimrao Ambedkar University. The topic of my presentation is microarray data analysis, advantages and limitations. So this is the table of content, introduction of the DNA microarray, history, types, working applications, advantages, limitations and conclusion. So first of all, let us understand about DNA microarray. As all the cells in the human body contain identical genetic material, but the same gene are not active in every cell. Studying which genes are active and which are inactive in different cell types helps scientists to understand both how these cells function normally and how they are affected when various genes do not perform properly. In past, scientists were able to conduct these genetic analysis on a few genes at once. With development of DNA microarray, however, scientists now examine how thousands of genes express and their products interact at any time. Microarray technology is a general laboratory approach that involves binding an array to thousands to millions of known nucleic acid fragments to a solid surface, referred as chip. The chip is then bathed with DNA or RNA isolated from a study sample, such as cell or tissue. It is an orderly arrangement of thousands of identified genes fixed on a solid support, usually glass, silicon chip, or nylon membrane, for the purpose of expressing, proliferating, monitoring expression level for thousands of genes simultaneously. The most well-known use of DNA microarray is the profiling mRNA level. However, DNA microarray have also been used to detect DNA protein interaction, alternatively spliced variants, the epigenetic status of the genome. So, the invention of microarray technology. So the invention of microarray technology in 1980s, microarray was proposed as a means of studying gene expression. Southern blotting was also invented, but in southern blotting, a specific number of DNAs sequences can be detected. Eventually, these assays can be miniaturized to allow thousands of genes to be analyzed simultaneously. So first of all, Fodder's work on oligonucleotide array. The first DNA microarray technology was developed by Stephen Fodder and his team from the FMX Research Institute. Photolithography allowed them to synthesize thousands of different oligonucleotides on the surface. Then, after that, the invention of cDNA microarray was pioneered by Patrick W. Brown and Ronald W. Davis. So, after commercialization and further refinement, microarray was made easily accessible when commercialized by Affymetrix. Gene chip microarray was developed for generating high density oligoarray using photolithography process. Hence, this created room for other firms to access the same market, thus improving the performance. Three types of DNA microarray. The first includes the glass DNA microarray. It involves the microspotting of prefabricated cDNA fragments on the glass slide. It is fabricated either by pen tip deposition or inkjet deposition. High density oligonucleotide microarray, often referred to as a chip, which involves in situ oligonucleotide synthesis. They are produced by chemically synthesizing of the oligonucleotide directly on the coated quad surface. This technology allows very high spot densities. cDNA microarray, it was invented in 1995. The invention of cDNA microarray was by Patrick W. Brown and Ronald Davis from the Stanford University. For this, they employed PCR on the synthesized cDNA and then transferred it onto a hard substratum. The working of DNA microarray. So the working starts with the microarray analysis includes the following step. First is the preparation of sample. The extraction of RNA include isolation of RNA from the sample, for example, cells or tissue. This RNA is now converted into complementary DNA by reverse transcriptase. Probe design and its labeling. Complementary sequences of the targeted gene are designed and labeled with the fluorescent dye. After that, the construction of array. The probes prepared earlier are now immobilized onto the solid surface for the formation of array. Hybridization of cDNA with the probes on the array occurs. Array is then scanned for the detection of fluorescent signals from labeled cDNA. The intensity of fluorescence are analyzed at each spot on the array. The data is analyzed and the identified gene expression pattern are interpreted. So applications of microarray. Applications of microarray includes the investigating cellular stage and process. Patterns of expressions that change with the cellular state or the growth condition can give clue to the mechanism of process or the changing from aerobic to anaerobic metabolism. Second is diagnosis of disease. Testing for the presence of mutation can confirm the diagnosis of suspected genetic diseases, including detection of a late onset condition such as Huntington's disease. Drug selection allow detection of genetic factors that govern responses to drugs, that some patients render treatment ineffective and in other cases unusual serious adverse reactions. Fourth is the specialized diagnosis of diseases. Different types of leukemia can also be diagnosed with the help of this technique. Pathogen resistance, comparison to genotype of expression patterns between bacterial strains susceptible and resistant to an antibiotic, point to protein involved in the mechanism. And the sixth is investigating cellular state during the cell cycle. The advantages of microarray techniques involve the use of microarray techniques include various advantages in the present as well as will be of greater use in the future. So first is the higher input. Higher input microarray allows researchers to get higher input with low higher output with the low input and lesser information. As in traditional technique, one gene is analyzed at a time. It requires more time as compared to microarray. Second is genome-wide expression, providing a comprehensive view of gene expression across the entire gene. Identification of gene association with specific biological processes, diseases, and conditions. Discovery of biomarkers. It helps in discovery of potential markers associated with diseases, disease higher throughput. DNA microarray analyze thousands of genes simultaneously. It is highly sensitive and it is highly specific. The results are very fast and it can analyze large amount of samples at a time. And it is relatively low cost than other techniques. That's why it is preferred more. It is a standard approach for data submission. So limitations. The limitations include limited dynamic range. They may not accurately detect extremely low or high level of gene expression. Cross-hybridization can lead to false positive signals impacting the accuracy. Sensitivity and specificity in microarray. Some probes are more sensitive than the other, so may not be able to detect transcriptions that are present in low amount. Depending on prior information, microarray relies on the existing genomic information. Inability to detect alternative splicing. So microarray may not be able to detect alternatively splicing, limiting the analysis of transcription isoforms. Biological complexities, post-transcriptional modifications and protein-protein interactions are not easily captured, creating issues. Data analysis challenges, inappropriate normalization methods or statistical tests may lead to misinterpretation of result and cost considerations. While microarray are often cost effective for large scale experiment, the cost associated with probe design, array fabrication and equipments can be significant. Conclusion. As a conclusion, microarray technique may help us understanding gene expression patterns and molecular mechanisms of various biological processes. It has great contribution to gene expression, proliferating biomarkers, etc., biomarker discovery, etc. The limitations include sensitivity, specificity, and careful consideration. Moreover, its cost effectiveness is the greater advantages for researchers. The further of genomic analysis lie to embrace a combination of technique that suits best for specific goals of study. These are my references. Thank you.
Next presentation, Nancy Yadav. Nancy.
Yes, sir.
Present your PPT.
Nancy Jadad, PG scholar, right?
No.
PhD?
PG, MSc.
PG. Okay, okay. Nancy. Nancy, please present your paper. Sir, next.
Next, Prerna Singh. Prerna Singh.
Prerna
Yes,
Singh,
sir.
please present.
So is it visible?
Yeah, it's visible.
Hello?
Yeah, it's visible.
Yes. Okay, sir. Good afternoon, everyone. My name is
No,
Prerna Singh.
share your
I'm
PPT.
pursuing
This is your
MSc.
screen. Your screen is visible, but your PPT is not visible.
Okay, sir. Just a minute.
You can go share, then window, and then just click PPT.
So is it visible now? Hello?
Yes.
So
No,
is it
no, no, it's
visible now?
not visible. No, not visible.
Okay, sir. Okay, sir. Just a minute. So is it visible now? So is it visible now?
Yes. Yes.
Yes, yes, it's visible.
Good afternoon, everyone. My name is Prerna Singh. I'm pursuing MSc Zoology in Baba Saheb Bhimrao Ambedkar University, Central University, Lucknow. My topic is
Role of
role of
cutting
cutting edge
technologies in medically
technologies in
important
medically
nematodes of
important
India.
nematodes of India.
As we know that
As we
parasites
know
are
that parasites
around
are
1 to 2
around 1 to
billion
2 billion. They are causing infections, which leads to the death of the
organisms.
organism.
Parasites are
Parasites are
eukaryotes
eukaryotes, and
and
hence they share various molecular and biochemical properties
There are certain compounds which damage the
Please
DNA
connect in only
and
one
these
device.
damaged DNA these damaged
DNAs
by the
are
DNA.
repaired by the DNA repair.
There are various cutting-edge technologies for trematodes being used, like mass drug administration program, herbal medicine, mRNA vaccine technology, PCR technology, CRISPR technology, and nanotechnology. So first is mass drug administration program. These programs are carried out in the world for control of liver flukes mainly. The praziquantel drug is used for treating many trematode infections. Its dosage is based on the population's weight to ensure proper administration. The successful mass drug administration programs are carried out in various places, and they involve the educating communities about the importance of treatment, transmission cycle of the parasites involved, and various hygienic practices so that the reinfection can be prevented. There are certain assessing drug efficacies which are provided to monitor the infection of the parasite. The second is herbal medicine. From ancient times, as we know that our ancestors were using the plants present in the jungle or places where they live, and they used its extract and byproducts to treat various parasitic infections. Some medical plants like Melissa officinalis, Citrus limon, Thymus vulgaris, they show anti-parasitic properties. Because of not knowing these side effects of these plants, their limitation, they are very limited in use. Their leaves, flowers, or roots of the plants are being directly used for their treatment. They target the genome sequence directly, and they cause the mutations which ultimately lead to the death of the trematodes. The next is mRNA vaccine technology. The developing vaccines for trematode presents various unique challenges due to the complexity of their life cycles and their way of interacting with the host immune system. The vaccine development here focuses on identifying suitable antigens that can stimulate an immune response. And this would lead to the permanent cure of the various trematode infections. It will be that much effective. The various epitopes are which are present on the antigen, they will be recognized by the immune system, and these will act against the trematode infection. The next is our PCR technology, which is various widely used in the study of trematodes for so many of purposes. They such as like species identification, genetic characterization, and diagnosis. It implies the genome regions of trematodes. It helps in the understanding the genetic diversity, the population structure. It has a qPCR, which is known as quantitative PCR. It is very useful in detecting the severity of the infections. The next is a CRISPR technology. Here CRISPR refers to the clustered regularly interspaced short palindromic repeats, and it is commonly known as CRISPR-Cas9. The CRISPR technology has transformative potential in study and control of the trematodes. The CRISPR-Cas9 allows the precise modification of some trematode genomes. It will help in the identification and certain modifications of the antigens for various vaccine development against the trematodes, which will produce effective targeted vaccines. For this, the CRISPR uses the RNA molecules. It is very accurate in DNA cutting, and it manipulates the genetic information within the organisms and its host. The next is the nanotechnology. The nanotechnology requires the nanoparticles, and these nanoparticles interact with specific trematode molecules, and which enables the precise diagnostic and imaging. The nano-based drug delivery system enhances the efficiency and targeted delivery of anti-trematode drugs, and this nanotechnology develops the sensitive biosensors for detecting the trematode infections. The nano-sized materials can be employed to create various platforms for vaccine delivery, enhancing the efficacy of the vaccines against the trematode parasites. They induces a cytotoxic effect on the cell of the host and the organism. There are various advantages of this nanotechnology, like it is very eco-friendly, it is cost effective, it is a very simple, so it does not require the expertise. So I want to conclude my presentation on saying that there are various diverse approaches which can produce the advancements in the parasitic disease management, which emphasizes the ongoing need of research, collaboration, and innovation in the quest of effective solutions. These parasitic diseases require the multi-approach of combining the traditional methods with the cutting-edge technologies. These are my references. Thank you very much.
Thank you, Prerna. Next participant, Asteria. Asteria Sharma. Asteria Sharma, Bikram.
Sir, hello.
Hello, please present your paper.
Wait a minute, sir. Screen. Hello.
Hello.
Sir, please screen.
Yeah, please.
A very good afternoon to everyone. My name is Vikram Singh and I am here to deliver my presentation on topic nanofluid implementation in porous media. First of all, the content is first is introduction, then porous media, then its application, some mathematical models and literature review, and in the last, conclusion. First is introduction. Nanofluids, a groundbreaking advancement introduced by COI in 1995, are sophisticated working fluids enriched with nanoparticles. Incorporating nanoparticles into base fluids substantially improve thermal and heat transfer characteristics. Metal oxides, nitride ceramics, and carbon ceramics stand out as preferred nanoparticle choices. Here is the representation of preparation of nanofluid. We can prepare nanofluid by mixing nanoparticles and base fluid, where base fluid are most commonly base fluid are water, ethylene glycol, glycerin, paraffin wax, and etc. And mixing them, we can have nanofluid. Next is porous media. Porous media is basically a material with pores through which the fluid flow during its motion. Porosity defining the void space as a percentage of the total volume in the porous medium. Directly influence porous storage, passage, and enhance permeability, thermal conductivity, and heat transfer rates. The combined influence of nanofluids and porous material in elevating the transfer rate of heat has supported considerable research into the thermal attributes of nanofluid flow within the porous media. Next are some application of porous media. First one is thermal management in electronics. In electronic designs, incorporating porous heat sink with nanofluids containing high thermal conductivity nanoparticles like graphene enhance thermal management. The porous structure facilitates efficient heat dissipation, optimizing overall cooling efficiency for devices such as laptops and smartphones. Second one is radiator design for engine cooling. In automotive engineering, integrating porous radiator with nanofluids featuring high thermal conductivity nanoparticles like aluminum oxides improving engine cooling. This approach enhances heat dissipation, ensuring optimal thermal regulation, preventing overheating, and extending the overall efficiency of the vehicle's engine. And the third one is improving the efficiency of solar thermal collector for renewable energy generation. Nanofluids with sunlight absorbing nanoparticles can flow through porous absorber in the solar collectors. The porous structure enhances fluid flow, while the nanoparticles increase the absorption of solar radiation, improving the overall efficiency of converting sunlight into heat for energy application. There are various other applications of porous media with nanofluid in diverse fields. Next is correlation between nanofluids and porous media. There are basically three base models that are used for defining the correlation between nanofluids and porous media. First one is Darcy model. Darcy model is used to describe fluid flow through porous media. It states that velocity of fluid flow through porous media is directly proportional to pressure gradient. This model assumes steady state laminar flow and does not consider factors such as fluid compressibility or thermal effects. Second one is Forsheimer model. It is an extension of Darcy model by adding Forsheimer term that is proportional to square of fluid velocity and captures non-linear effect at high flow rates. This model is particularly useful in scenarios with porous media involving higher velocities. Third one is Brinkman model. This model is significant, particularly in the situation where both viscous and inertial effects are significant. This model extends Darcy model by introducing additional term for inertial effect. Here are some literature review which define the use of Darcy model, Forsheimer model, and Brinkman model for representation of porous media involving with nanofluids. And here are some conclusions. From the literature survey, it has been observed that Darcy model is least used model because it works in the specific case of slow viscous flow. Second, water and ethylene glycol are found mostly used working waste fluids for nanofluids. And third one is copper and silver are seen mostly used metal due to their superior thermal conductivity and compatibility with various fluid systems. Thank you.
Thank you, Mr. Vikram. Next participant, Bansikha. Next participant, Bansikha.
Yes, sir.
Please present your paper.
Okay, sir.
Okay.
So, my presentation, which is also
No,
important
Ankit
role
Kumar.
Important role of the
Bansikha, please present your paper.
Oh, okay, sir. Sir, is it visible?
Yeah, it's visible.
Hello, am I audible, sir?
Yes, you are audible.
My name is Vanshika. A very good afternoon to everybody present here. I'll be presenting on the topic study on epigeic earthworm species and its gut microbes on degradation of synthetic dyes. So let's have a brief introduction to the dyes. So dyes are widely used in industries like textile, leather, paint, food, etc. 25 types of dye groups are well known based on their chemical chromophore structures. They are generally derived from naphthalene and benzene, which act as their raw material. And dyes are highly soluble in water, owing to which it is very challenging to remove them. So today, dye manufacturing industries are known to be primary source of water pollution. Various methods are employed like physiochemical, biological methods, but these methods are highly insufficient. So mainly biological methods which involve use of algae, yeast, bacteria, and fungi, they have capability to mineralize and decolorize various dyes. So here is some background information on earthworms and how they can play a role in dye degradation. So earthworms are mainly known as the soil engineers. They help in building and maintaining the soil structure and take part in nutrient cycling through mineralization and humidification. They also serve as bioindicators by indicating the level of soil contaminants such as agrochemicals, heavy metals, toxic substances, and industrial effluents. The microorganisms present inside the earthworm gut are the main driver of their activities. These microbes are generally obtained from the soil surrounding these earthworms and the organic matter which is present in the soil. So earthworms, they are being used as bioagents for rapid and efficient decomposition and remediation of various industrial and organic wastes. Synthetic dyes and its types. So dyes are classified on the basis of their structure and application. Various dyes like azo dyes, nitro dyes, phthalate, triphenyl methane dyes, indigo dyes, and anthraquinone dyes are known today. Out of these dyes, azo dyes and anthraquinone dyes are two most widely used ones. Azo, it consists of an azo bond, and it belongs to class of heterocyclic and aromatic compounds, which are also known for their carcinogenic properties. Here we can see the structure of azo dyes. Some of their examples are acid orange, acid red, methyl orange, etc. Anthraquinone is the second most widely used dye because of its easy accessibility, low price, but it is known to be highly toxic to humans and microorganisms. This is the structure of azo dye. It consists of an anthraquinone chromophore group, which includes a benzene ring with two carbonyl groups on both sides. As per the environmental impact of these dyes, which are released from industries, they cause an increase in BOD and COD levels, which also inhibits the photosynthetic activity of organisms. Heavy metals present in textile industry effluents are not biodegradable and hence accumulate in primary organs in body, and over time they begin causing various diseases. In humans, they are known to cause respiratory and nasal symptoms, for example, asthma, rhinitis, dermatitis, etc. So why the biological degradation is needed? There are various physiochemical and other processes like absorption and membrane filtration, which also help in dye degradation. But the main disadvantage of these processes is that they produce a large amount of sludge and secondary waste streams which need further treatment. So in conclusion, these processes are highly inefficient, whereas it is known that biological degradation of pollutants is eco-friendly and complete mineralization of organic compounds with low sludge generation happens. So these are few microbes which are involved in dye degradation. Bacteria and algae, they are known to help in degrading dyes by production of a reductase enzyme, also known as azoreductase. And